Hi, this is Amanda, and today we're going to talk about the cell membrane and its structure and function in the body. The cell membrane is composed of a phospholipid bilayer with heads and tails. The heads are hydrophilic and the tails are hydrophobic. Hydrophilic means that they are water-loving and they face outwards towards the water. The tails are hydrophobic because they are water-fearing. The cell membrane is known as a fluid mosaic model because each lipid is fluid in that it can move, and it represents a tile in a mosaic. The lipids are fluid in that they can move laterally, sometimes jumping over one another to get to a new place in the membrane. Membranes are composed of many proteins, including peripheral proteins on the outside of the bilayer and integral proteins embedded in the lipids. Another important protein is a channel protein, which creates a tunnel through the membrane to allow certain particles to get in the cell. The fluid mosaic model and the function of proteins causes the cell membrane to be selectively permeable, which means only certain particles can enter the cell. The cell membrane's function is to move particles in and out of the cell, and one way it does this is by using passive transport. The cell membrane's goal is to have the cell in an isotonic solution where there is equal particle concentration inside and outside of the cell by dispersing water evenly to both sides. The cell membrane can exist in a hypertonic solution when there's a higher particle concentration outside the cell, in which case the cell must shrink and the water moves to the outside of the cell to even out the particles. The cell can exist in a hypotonic solution when there are more particles inside the cell than outside, so water from the solution moves into the cell, causing the cell to swell with water. Another type of diffusion is called facilitated diffusion, in which particles are transported by a channel protein through the cell membrane. This requires no energy. Another type of diffusion is called active transport, in which ATP, or energy, is used to transport particles. ATP is used to power the protein that transports the particles to and from the cell. Another form of particle transport by the cell membrane is in the form of exocytosis, in which a Golgi body moves particles into a vesicle. Then the vessel moves to the cell membrane and releases the particles outside of the cell. This is called exocytosis. The cell can also perform endocytosis, in which particles from the outside of the cell are taken in by a vesicle and moved to the Golgi body. Endocytosis has three kinds, which include phagocytosis, in which the cell is eating, penocytosis, in which the cell is drinking, and receptor-mediated endocytosis.
Hi, this is Morgan, and today we're going to be taking a tour of the eukaryotic cell. Prokaryotic cells belong to domains archaea and bacteria, while eukaryotic cells belong to protists, fungi, animal, and plants. Prokaryotic cells do not have a nucleus, they have no membrane-bound organelles, and their DNA is located in the nucleoid. Eukaryotic cells do have a nucleus, do have membrane-bound organelles, and their DNA is in the nucleus. The flagellum is an extension of the plasma membrane that is responsible for locomotion in some animal cells. The centrosome is the region of the cell where the microtubules are initiated. Microvilli are tiny projections on the outside of the cell that increase the surface area. The nucleus is the center of information in the cell and has one or more nucleoli. It contains most of the cell's genes, which are organized into chromosomes made of chromatin. The nuclear envelope is a double membrane that encloses the nucleus. It is perforated by pores and continuous with the endoplasmic reticulum. The nucleolus is involved in the production of ribosomes. Chromatin is the material consisting of DNA and proteins, which are visible as chromosomes when the cell is dividing. The central vacuole is responsible for the storage and breakdown of waste products and the hydrolysis of macromolecules. Enlargement of the vacuole is evidence of plant growth. The plasmodesmata are channels that go through the cell walls to connect cytoplasms of adjacent cells. Ribosomes are protein factories that are either free in the cytosol or bound to the rough ER or nuclear envelope. Both are structurally identical and can alternate between the two roles. The mitochondria is where cellular respiration occurs and most ATP is generated here. Some cells have one large mitochondria, but most have hundreds or even thousands. The Golgi body is associated with shipping and receiving because the products of the endoplasmic reticulum are modified and stored here and then sent all throughout the cell. Lysosomes digest macromolecules, carry out digestion throughout the cell, and recycle the cell's own organic material. Digestion occurs through phagocytosis, when amoebas and other protists and some human cells engulf the food. The smooth ER is responsible for the synthesis of lipids such as oils, phospholipids, and steroids, the metabolism of carbohydrates, and the detoxification of poisons. In some cases, it also is responsible for storing calcium ions. The rough ER secretes proteins that are produced by the ribosomes attached to it. The endoplasmic reticulum is a biosynthetic factory that accounts for more than half of the total membrane in eukaryotic cells. It contains the rough ER and the smooth ER. Endoplasmic means within the cytoplasm, and reticulum means a little net. The three types of vacuoles are food, contractile, and central. Food vacuoles are formed by phagocytosis to be used as food for the cell. Contractile vacuoles help move excess water out of certain freshwater protists. Central vacuoles were just mentioned. Peroxisomes are responsible for oxidation and various specialized metabolic functions. They are bounded by a single membrane and produce hydrogen peroxide that convert to water. They also import proteins from the cytosol. Glycoproteins are proteins that have one or more carbohydrates covalently bonded to them. Vesicles are sacs made of membrane in the cytoplasm. Transport vesicles are vesicles that are in transit, carrying molecules from one part of the cell to another. Cristae are infoldings of the inner membrane of the mitochondria. They house electron transport chains and ATP synthase enzymes. Chloroplasts are photosynthetic organelles that convert solar energy to chemical energy. Inside, they contain thylakoids, granum, and stroma. Thylakoids are individual flattened sacs inside the chloroplast. Granum are stacks of thylakoids inside the chloroplasts. They function in the light reaction during photosynthesis. Stroma is the fluid inside the chloroplast. It is involved in the synthesis of organic molecules using carbon dioxide and water. The organelles specific to animal cells are lysosomes, centrosomes that have centrioles, and flagella, although some flagella are present in plant sperm. The organelles specific to a plant cell are chloroplasts, a central vacuole, cell wall, and plasmodesmata. The cytoskeleton consists of microfilaments, intermediate filaments, and microtubules. Together, they reinforce the cell shape and function in cell movement. They are all made of proteins. The plasma membrane is the membrane that encloses the cell. The cell wall is the outer layer of the cell that maintains the shape and protects it from outside damage. It is primarily made of cellulose, polysaccharides, and protein. The mitochondrial matrix is the part of the mitochondria enclosed by the inner membrane, which contains enzymes and substrates for the citric acid cycle. The endomembrane system is the collection of membranes throughout the eukaryotic cell and contains the rough and smooth ER, the Golgi apparatus, lysosomes, and vacuoles.